So, welcome to Excellence Icon, and I hope so that you all are doing very well. Today, we are starting the chapter number 5 of your AS Biology 9700, the cycle or the mitotic cell cycle. So, what does this mean? Basically, it is a cycle of the mitosis, as you have learned in your O levels about the meiosis and mitosis. So, this is the cycle of the mitosis where what involves is being expressed. All right. So, starting from I am having the two types of PDFs of mitotic cycles. So, I'll be making you reading and understanding from these boards. So, it might be very helpful for you. The cell cycle can of us of as the life cycle of a cell. In the other words, it is a series of growth and development steps a cell undergoes between its birth, formation by the division of a mother cell, and reproduction, division to make two new daughter cells. It means that in mitosis, two new daughter cells are made, and in meiosis, four new daughter cells are made. So, what is interphase? Let's enter the cycle just as a cell form by division of its mother cell. It is a single parent, alright? What must this newborn cell do next if it wants to go on and divide itself? Preparation for division happens in three steps, and the it means that we have been talking about the interface. In the interface, there are three steps: G1 phase, and then S phase, and then G2 phase. So, what does this means? Okay, G1 phase. During G1 phase, what does G means here is growth, alright, or we can say it as gap, also called the first gap phase. Here represents the gap, and in this the growth basically occurs as the cell grows physically larger copies organelles. What does organelles means? The mini or the tiny organs, such as the ribosomes and whatever we'll be learning in the cell structure, and makes the molecular building blocks it will need in later steps. Do cells always grow before they divide? Alright, so this is the question, just leave it, and after this I'll be making you understand so that now you can answer these questions which have been written in the bracket. It might be very helpful for you for in your AS, but first of all learning the chapter. In the S phase, what does the S means is synthesize. What is the synthesis of DNA? In S phase, the cell synthesizes a complete copy of the DNA in its nucleus. It also duplicates or replicates a microtubule organizing structure called the chromosomes. Oh, sorry, centromeres, centrosomes. So the centrosomes help separate DNA during M phase. M phase is basically the mitotic phase. Alright, so the centrosomes. What does the centrosomes means? These are the structures. I'll be making you understand from the diagram, so it could be more clarified to you that the centrosomes are the structures, microtubule organizing structures. They form the microtubules. Okay, in the G2 phase. Alright, so that was of S phase where the DNA replicate. Understand? When there was G1 phase, which is the interphase first step, in the G1 phase, there was a growth of the organelles. At the S phase, there was the DNA replication. At the G1 phase, it decides either to duplicate or not. So when they decides to replicate, so then they goes towards the S phase where they duplicate or replicate the DNA. Alright, so the DNA is replicated in the S phase. In G2 phase, during the second gap phase or G2 phase, the cell grows more, makes proteins and organelles, and begins to reorganize its contents in preparation for mitosis. G phase ends when mitosis begins. It is the last stage of interphase. Alright, so G1 and S and G2 phases are the phases of interphase. So this is all happened about the growth and then the replication of DNA, and then G2 phase where they also have to make the proteins and organelles. All right. The G1S and G2 phases together are known as interphase. The prefix inter means between, reflecting that interphase takes place between one mitotic or M phase and the next. All right. So this is the most important figure and the cycle very essential for you guys in your mitotic cycle. As you can see here that this is the G1 phase, and you can also observe that the interphase is having a lot of ratio in this pie chart. As you can see that there's G1 and then S and then G2 having a lot of sites, while the mitosis and the cytokinesis is having a lesser site. So it means that the interface is essential for you to understand where the G1 or the gap 1 phase and the gap 2 phase of the interface are having the cell growth simply, while in the S phase, S means synthesis of DNA, meanwhile the DNA replicates. Alright, in the mitosis, it is a mitotic phase where the formation of two daughter cells and then cytokinesis. What is cytokinesis? We'll be understanding in this video. So this is very essential for you. First of all, G1 phase. Once it enters to the G1 phase, has been working making more organelles and growing the cells. It is the phase which decide for the cell either to replicate or not. If the signals from the brain came to the cell to duplicate, so then it enters to the S phase of interphase where the DNA replicates first, and then G2 phase where the cell growth occurs, and then the mitosis where the cell divides into two uh, daughter cells. Alright, so at the M phase, during the mitotic phase, the cell divides its copied DNA and cytoplasm to make two new cells. M phase involves two distinct division related processes: mitosis and cytokinesis. Cytokinesis means the division of cytoplasm. Alright, and mitosis means the division of cell. In mitosis, the nuclear DNA of the cell condenses into visible chromosomes and is pulled apart by the mitotic spindle, a specialized structure made out of microtubules. Mitosis takes place in four stages: prophase, sometimes divided into early prophase and prometaphase, metaphase, anaphase, and telophase. Now, what does this mean that in mitosis the nuclear DNA of cell condenses into visible chromosomes? It means the word condenses means that the DNA joins together and the DNA become more visible, while the chromosomes become the DNA more visible, and that's why it is visible through the electron microscope. All right. So this is how we see that how the chromosomes are basically dividing. We'll be also understanding the structure of the chromosome, which is very essential before starting this stages, but we'll be understanding in the next PDF. And is pulled apart by the mitotic spindle. So the mitotic spindle is formed. How it is formed? It is formed by the centrosomes. By the centrosomes, the mitotic spindles are being formed, which carries out the uh, visible chromosomes and pull apart to each other so that the two daughter cells have this identical modification, a specialized structure made out of microtubules. All right. You can learn more about these stages in the video of mitosis. In cytokinesis, the cytoplasm of a cell is split into making two new cells. Cytokinesis usually begins just as mitosis is ending, with a little overlap. Importantly, cytokinesis takes place differently in animals and plant cells. How is it so? This is the reason that as the animals having this cytoplasm, so in the cytokinesis in animals, the cytoplasm is dividing and the cell membrane basically divides. But in plant cell, does it have anything extra? Cell wall. It means that the cell wall in plant cells divide. So cytokinesis in plant cell means the cell wall is basically dividing, while it is absent in animals. All right. What happens to the two daughter cells produced in one round of the cell cycle? Okay, this depends on what type of cells they are. Wait a minute. All right. So this depends on what type of cells they are. Some types of cells divide rapidly, and in these cases, the daughter cells may immediately undergo another round of cell division. For instance, many cell types in an early embryo divide rapidly, and so do cells in a tumor. Other types of cells divide slowly or not at all. These cells may exist G1 phase, as I told you, and enter a resting state called GO phase. G0 phase. Okay. In G0, a cell is not actively preparing to divide. What I say you to? Uh, what I say to you, that is basically when the brain give the signal to the cell not to divide, so then it go towards the G0 phase. So this is just for the information, is not part of your syllabus, but it's just for the information. Alright, so for instance, it might conduct signals as a neuron like the one in the drawing below, or store carbohydrates in the liver cell. G0 is a permanent state for some cells, while others may restart division if they get the right signals. So this is what basically that how long does the cell cycle takes, okay? Different cells take different lengths of time to complete the cell cycle. A typical human cell might take about 24 hours to divide, but just wait a minute. Okay. But fast cycling mammalian cells, like the ones that line the intestine, can complete a cycle every 9 to 10 hours when they are grown in culture. Different types of cells also split their time between the cell cycle phases in different ways. In early frog embryos, for example, cells spend almost no time in G1 and G2 phase and instead rapidly cycle between S and M phases, resulting in the division of one big cell, the zygote, into many smaller cells. What is mitosis? Mitosis is a type of cell division in which one cell, the mother, means only one single parent cell, divides to produce two new daughter cells, which are genetically identical to itself. In the context of the cell cycle, mitosis is the part of the division process in which the DNA of cell's nucleus is split into two equal sets of chromosomes. Alright. So where were we? The great majority of the cell divisions that happen in your body involve mitosis. During development and growth, mitosis populates an organism's body with cells, and throughout an organism's life, it replaces old, worn-out cells with new ones. For single-celled eukaryotes like yeast, mitotic cell divisions are actually a form of reproduction, adding new individuals to the population. In all of these cases, the goal of mitosis is to make sure that each daughter cell gets a perfect full set of chromosomes. If the number of chromosomes are not equal, it means that there is abnormal cases. Cells with too few or too many chromosomes usually don't function well, they may not survive, or they may even cause cancer. In this chapter, cancer is also important. So, when cells undergo mitosis, they don't just divide their DNA at random and toss it into piles for the daughter cells. Instead, they split up their replicated chromosomes in a carefully organized series of steps. Phases of mitosis. Mitosis consists of four basic phases: prophase, metaphase, anaphase, and telophase. Some textbooks list five, breaking prophase into an early prophase called prophase and a late prophase called prometaphase. These phases occur in strict sequential order and cytokinesis. Okay, the process of dividing the cell contents to make two new cells starts in anaphase and telophase. So this is the cycle of mitosis as prophase, metaphase, anaphase, and telophase, where the anaphase and telophase occurs cytokinesis. It is late G2, means late gap 2 phase. Now you can observe here the centrosomes. Alright, and the centrosomes consist of centrioles. These are the basic centrioles which have a lot of microtubules forming this mitotic spindle. And there's the nucleolus in the nucleus, and there's the nuclear membrane and DNA which is replicated. Means now as you are seeing that there are two lines of each DNA, it means that it's replicated. Let's start by looking at the cell right before it begins mitosis. This cell is in interphase late G, all right, and has already copied its DNA. So the chromosomes in the nucleus each consist of two connected copies called sister chromatids. You can't see the chromosomes very clearly at this point because they are still in their long, stringly decondensed form. This animal cells has also make a copy of its centrosome, an organelle that will play a key role in. Okay, wait a minute. All right. This animal cell has also make a copy of its centrosome, an organelle that will play a key role in orchestrating mitosis. So there are two centrosomes. Plant cells generally don't have centrosome with centrioles but have a different type of microtubule organizing center that plays a similar role. This is the early prophase, as where you can see that the mitotic spindle starts to form and now the centrosomes are traveling to the opposite sides or the opposite poles. Alright, chromosomes start to condense and nucleolus is gone, or we can say that this disappears. In early prophase, the cell starts to break down some structures and build others up, setting the stage for division of the chromosomes. The chromosomes start to condense, making them easier to pull apart later on. The mitotic spindle begins to form. The spindle is a structure made of microtubules, strong fibers that are a part of the cell's skeleton. Its job is to organize the chromosomes and move them around during mitosis. The spindle grows between the centrosomes as they move apart. The nucleolus or plural. Okay. A part of the nucleus where ribosomes are made disappears. This is the sign that the nucleus is getting ready to break down. And this is the late prophase or prometaphase. You can observe that the centrosomes are as the centrosomes are at the side of the. Alright, so this is the centrosomes at the side of the opposite poles, forming the microtubule, and here's the nuclear envelope, which is breaks down. Okay, so the nuclear envelopes basically break down, which you can now see from this line of the green. The chromosomes fully condensed, which are now visible. Okay. In late prophase, sometimes also called prometaphase, the mitotic spindle begins to capture and organize the chromosomes. The chromosomes become even more condensed, so they are very compact. The nuclear envelope breaks down, releasing the chromosomes. The mitotic spindle grows more, and some of the microtubules start to capture chromosomes. This is the spindle anatomy, where you can see what are the centro. Okay, here's the centromere. The centromere is, we'll be understanding in the chromosomes structure, that this holds up the chromatids. It holds up the chromatids to form the chromosome. And this is the kinetochore. This is the kinetochore, which is basically a type of a solid, or what you can say is a part which joins basically the centromere to the spindle. And there's the kinetochore, okay, and microtubule, okay. Microtubules can bind to chromosomes at the kinetochore, a patch of protein found on the centromere of each sister chromatids. What are the sister chromatids? We'll be understanding in the structure of chromosomes. Okay, so then this centromeres are the regions of DNA where the sister chromatids are most tightly connected. Microtubules that bind the chromosomes are called kinetochore microtubules. Okay, so microtubules that don't bind to kinetochores can grab on the microtubules from the opposite pole, stabilizing the spindle. More microtubules extend from each centrosome towards the edges of the cell, forming a structure called ester. All right. And this is the metaphase. In the metaphase, you can see that the chromosomes line up at metaphase plate. And this is essential to line up in the equal. So you can see that what is happening in the anaphase. The anaphase will be making you understand about that how does this works. In metaphase, the spindle has captured all the chromosomes and lined them up at the middle of the cell, ready to divide. All the chromosomes align at the metaphase plate, not a physical structure, just a term for the plane where the chromosomes line up. At this stage, the two kinetochores of each chromosome should be attached to microtubules from opposite spindle poles. Before proceeding to anaphase, the cell will check to make sure that all the chromosomes are at the metaphase plate with their kinetochores correctly attached to the microtubules. This is very essential, as you can say that if it is not being investigating or not checking, so there must be the un there may be not the equal or unequal distributions of the chromosome, and this will be causing the abnormality, and then the cell will be obviously not function what it want to function, and this will be abnormals. All right. This is called the spindle point and helps ensure that the sister chromatids will split evenly between the two daughter cells when they separate in the next step. If a chromosome is not properly aligned or attached, the cell will uh, halt division until the problem is fixed. Alright, so moving towards the anaphase. In the anaphase, you can see that the microtubules push apart, and this how the chromatids are being separated and the equal distribution, so that this could be very identical to each other. The kinetochore microtubules pull chromosomes towards poles. In anaphase, the sister chromosomes separate from each other and are pulled towards opposite ends of the cell, and now the protein glue that holds the sister chromatids together is broken down, allowing to separate. Each is now its own chromosome. The chromosomes of each pair are pulled towards opposite ends of the cell. Microtubules not attached to the chromosomes elongate and push apart, separating the poles and making the cell long. The cell is nearly done dividing, and it starts to re-establish its normal structure as cytokinesis. The division of the cell contents takes place. The mitotic spindle is broken down into its building blocks, and the two new nuclei forms. one for each set of chromosomes, nuclear membranes and nuclei reappears. The chromosomes begin to decondense and return to their stringy form. As now you can see in the telophase, the chromosomes start to decondense. Okay, spindle disappears, nuclear membrane reforms, and the nucleus reappears. And this how this two identical daughter cells are being formed. Okay. Cytokinesis, the division of the cytoplasm to form two new cells, overlap with the final stages of mitosis. It may start in either anaphase or telophase depending on the cell and finishes shortly after telophase. In animal cells, cytokinesis is contractile, pinching the cell into two like a coin purse with a drawstring. The drawstring is a band of filaments made of a protein called actin. The pinch. Okay, so this is not the part of your syllabus. Okay, so this is the G1 phase where you can see. Okay, now this was. Sorry for that. I'll show you the diagram. Okay, so now after the telophase, again the G1 phase begins where the cytokinase and the two identical daughter cells formed. And now the G1 phase again starts, right? And this is how the cycle continues. When cytokinesis is finished, we end up with two new cells with a complete set of chromosomes identical to those of the mother cells. The daughter cells can now begin their own cellular lives, and depending on what they decide to be when they grow up, may may undergo mitosis themselves, repeating the cycle. And this is how you can say that these are the while in the plant, all the plants are identical because the asexual reproduction occurs and the mitosis occurs. While in the human beings, the meiosis and mitosis both occurs. While in the meiosis, the baby is having some features of the father and some features of the mother. I'm also having another PDF, but I'm not having more time. So as the time is going to be ended, I'll be soon making another video on the mitosis or the mitotic cell cycle, which I'll be reading to part 2, and then I'll be making understanding you the next PDF, which is also essential and helpful for you and may help you in your paper. So I hope so that you understand this. If you does not understand, you can comment me below. Until that, take care.